So as we discussed in the last video, there are two main groups of antigens. There are your ABO blood group antigens, which are coded for by one gene, and there is the RH blood group, which is coded for by a different gene. And if you have, let's say, the gene for the A antigen, you will have that A antigen, which is a marker on the outside of your red blood cell. And you will not have the antibodies that attack that antigen. We call them anti-A antibodies, right? So whichever antigens you have, you will not have those antibodies. And so you can test somebody's blood by adding the antibodies to the mystery blood, right? And if it's blood where I don't know what the blood type is, if I add anti-A antibodies to that blood and it clumps up, that means there must have been A antigens in that blood. So that person must have the A antigen. It must be type A or maybe type B, right? Type AB. So if I put the A antibody in there and it clumps, I have the A antigen. If I put the B antibody in there and it doesn't clump, there must not have been the B antigen in that blood. If I put the anti-RH or anti-D antibody in there and it does clump, that means they must have had the RH antigen or the D antigen. Same thing. So this person has the A and the RH antigens. They are A positive. They're positive for RH or D. If that makes sense. Now, in terms of transfusions, this is important to know because I cannot give my blood to anyone who has antibodies that have the same letter or match my antigens, or the antibodies will attack those antigens, as we talked about. So if I am A positive, meaning I have the A antigen and the RH antigen, I can't give blood, or I shouldn't give blood, to anyone who has the anti-A antibodies and or the anti-RH antibodies. So if I give blood to somebody, they better have the A antigen and the RH antigen, because that's the only way I can be sure that they don't have those antibodies, right? If you have an antigen, you never have the complementary antibody in your blood because it would attack your own body. So if I'm A positive, I can give to somebody else who's A positive. I can also give to somebody who's AB positive, because even though they also have the B antigen, they still have the A and the RH antigens, which means they won't have the anti-A or anti-RH antibodies. I can't give to anybody else or it'll cause an agglutination reaction. So remember, the donor's antigens and the recipient's antibodies are what cannot match up, right? So if I'm A positive and I'm the recipient, it matters about my antibodies and the donor's antigens, right? So what are my antibodies if I'm A positive? Well, anti-B, right? I can't have the A antibodies or the RH antibodies because they would attack my own cells. So I only have the B antibodies. So I can receive blood from anybody unless they have a B antigen, because then my B antibodies would attack them. So I can receive A positive blood or A negative blood. Actually, this is an error. A negative should be there as well. Or O positive or O negative, but not B and not AB. If there's a B there, you can't receive it. I apologize for that. This figure is straight out of your book. I'm hoping in the ebook they've fixed that by now, but I'll look into that with the publisher. So this should say A positive, A negative, O positive, O negative, as to who I can receive from if I'm A positive. Now, if you're AB positive, you can receive from anybody because you don't have any antibodies. You have all the antigens but no antibodies, so you're not going to attack any kind of blood, so you can receive from anybody. Conversely, if you're O negative, you have all the antibodies, right? You have no antigens, so you can give to anybody because no antibodies are going to attack you. So O negative is the universal donor of red blood cells. But you can only receive from other O negative individuals. Your blood type is coded for genetically. Right? You get this from your parents. And you have two alleles. If you have two A alleles, that means you make the A antigen, you're type A. If you have two B alleles, you make the B antigen, you're type B. If you have an A allele and a B allele, you make both antigens, you're type AB. If you have two alleles for O, that means neither of your alleles code for any antigen. You have no antigens, you're type O. Now, if you have a type A allele, which codes for type A antigen, and a type O allele, which codes for no antigen, you're type A. So essentially A is dominant over O, because you at least have one allele that codes for the A antigen. And if you are type A, you will have the anti-B antibodies from early in life, and it remains constant throughout life. But that's actually kind of abnormal. Typically, you do not make antibodies for an antigen until you've been exposed to it. So this is a difference that we see with the ABO blood group, in that you're born or very soon after birth, you're making those antibodies. For Rh, it's much more typical of the way that your normal antibody reactions work. So if you are Rh negative, you can have the anti-Rh antibodies, but you don't necessarily. It depends on if you've been exposed to the Rh antigen in the past. So maybe you've had some blood mixing at some point, and then you'll start making those anti-Rh antibodies. That will matter for something we're going to talk about in a moment, which is hemolytic disease of the newborn. Hemolytic disease of the newborn occurs when the baby or the fetus inside a mother, so this pre-birth, and the mother have conflicting blood types, right? And so this doesn't matter for the ABO blood group, because let's say you're type A and the baby's type B. Well, you make anti-B antibodies. Those could attack the fetus theoretically, but they're too big to cross the placenta. So those antibodies don't matter. But the anti-Rh antibodies are small enough to go across the placenta, and so they do matter. So in a case where the mother is Rh negative and therefore can make the anti-Rh antibodies, and the fetus is Rh positive, well, you could have an issue. Now, this generally doesn't matter for the first pregnancy when you have a Rh negative mother and an Rh positive fetus, because the Rh negative mother may not have been exposed to the Rh antigen before, and she probably won't be exposed during pregnancy. She could be, right? There may be some fetal blood cells that end up entering the bloodstream somehow, but generally the mixing doesn't happen until birth. But at birth, there's a lot of blood, and the placenta is being pulled away from the uterus after the baby is born, and it's a scene, and there is blood mixing. And usually when that first baby is born, mom is then exposed to that Rh positive antigen. So the next time Rh negative mom has an Rh positive fetus, her antibodies that she's making now will attack that fetus. And it takes mom a little time to build up those antibodies, but about 20% of Rh negative mothers who have Rh positive children get sensitized within 6 months of the delivery. So it doesn't necessarily happen, but it happens often enough to where it's a pretty big health issue. And if it happens, those antibodies from mom will move across the placenta into the fetal bloodstream and will attack the fetus's red blood cells, causing anemia. The red blood cell count will drop, and that can be fatal for the fetus without treatment. And this is a pretty good figure from your book showing that during your first pregnancy, again, if the mother is Rh negative and the developing infant is Rh positive, during the pregnancy, there's typically not much mixing of the blood, but there often is during the birth. And so the second pregnancy can cause issues. Again, the second pregnancy with an Rh negative mother and an Rh positive infant. If you have an Rh positive mother, this isn't an issue. If you have an Rh negative mother who has an Rh negative infant, also not an issue. So really this only matters when the mother is Rh negative and the father is Rh positive, then you have a chance of having an Rh positive infant. In order to prevent this, now there's a medication called Rogan that they shoot. It's this giant needle they shoot into you, and Rogan essentially it's like a fake antibody, and it binds to the antigens on the baby's blood cells before mom's immune system can bind to them. So it essentially masks them, so it keeps mom's immune system from realizing that there have been these antigens in her body, and so she never gets sensitized and ends up making the antibodies herself. All right, let's talk white blood cells. So white blood cells, as opposed to red blood cells, erythrocytes, are called leukocytes. They tend to make up a much smaller proportion of the blood cells that you have because they spend a short time in blood circulation, and they tend to then pop off into your tissues where infections can occur. They migrate out of the bloodstream. And you have several types. All of them are attracted by certain chemical stimuli, which essentially are like chemical alarm bells. So, the bat signal, right, is a visual signal that brings Batman in to help you take care of whatever criminal you're worried about, right? We don't have visual or light signals in your tissues, but you can release chemical signals that essentially say, hey, immune system, come help. And all of them are attracted by certain chemicals. Several of them, neutrophils, eosinophils, and monocytes, are capable of phagocytosis, which means the engulfing of a pathogen and eating it. Phagocytosis is cell eating. Monocytes specifically become macrophages, which literally means big eaters. And they become macrophages once they move out of the bloodstream, and they eat lots of stuff. We'll talk about them a bunch in the immune system chapter. There are two main groups of white blood cells, of leukocytes. Granular leukocytes have granules in them, little granulations that are essentially little balls of stuff that will be secreted out of the cell. And these include neutrophils, eosinophils, and basophils. Neutrophils being by far the most common kind of leukocyte you have, eosinophils and basophils actually being the two least common. In addition to granulocytes, you have two types of agranulocytes, meaning they don't have those big granules. If you look at them under a microscope, they look much smoother in the cytoplasm. And this includes monocytes, which, as I mentioned, become macrophages when they leave the blood, and lymphocytes. Now, lymphocytes are the second most common kind of leukocyte, and there are multiple types of lymphocytes. They're very important for your immune system and your adaptive immunity, and we'll talk about those a lot more once we get to the immune system chapter. Now, I do want you to recognize these types of white blood cells, right? So neutrophils look kind of like this, right? They have a multi-lobed nucleus. It's really weird. And they're granulocytes, so you can see little granules. Eosinophils, also a multi-lobed nucleus, but they're kind of reddish. I don't usually like to use color when I'm describing cells because it changes depending on the stain you're using, but otherwise they look a lot like neutrophils. That said, neutrophils are a lot more common, so if you're unsure, guess neutrophil. And basophils, we don't show one here, they're the rarest. You can't even see the nucleus because they have such thick granules. And they are important because they produce, in those granules, histamines. And histamines, you may be familiar with them, they cause inflammation. And you think of inflammation as a bad thing, but it's a way to dilate blood vessels to bring a lot more fluid to an area that flushes out toxins. It also brings lots of other white blood cells to an area to fight off something. Inflammation is a way that your body responds to an attack, and basophils are a big part of that. Now, you can have inflammation at inappropriate times, right? Things like allergies are inflammatory responses to things that you shouldn't be having inflammatory responses to, right? Pollen, right? Not attacking you. In that case, you can take an antihistamine that will block the action of these histamines. Antihistamines, fun fact, were developed first right up the road at the University of Cincinnati by George Revachol. The biology building today is named after him. He invented Benadryl. Monocytes are a type of agranulocyte. You can see it here. See a very smooth cytoplasm, and they have a large U-shaped nucleus. Those are monocytes. And lymphocytes are also agranulocytes, smooth cytoplasm, but they tend to have a big round nucleus in the middle, right? It takes up almost the entire cell there. In terms of function, lymphocytes are important for specific immunity. Monocytes become macrophages, which eat stuff. Basophils are important for inflammation. Eosinophils take down large parasites like worms and things that have been marked with antibodies, and neutrophils are general fighters that fight off a lot of bacterial infections.